Well, hello, everyone. It's really a pleasure to be part of this symposium, and thank you for your participation. I'm really delighted to tell you about how we're using this wonderful little model organism, a tropical fish called the zebrafish, to model some of the metabolic changes that may be occurring in ME. Before I begin, I'd just like to disclose my financial relationships and thank the funding agencies that have funded our work in our lab. Most relevant for this presentation is a gift from the Khosla family. We're deeply grateful for the financial support from the Khosla family that has really enabled all of the work that I'll be telling you about today. So my group at the University of Utah is one of 6 groups that are collaborating to study and work towards treatments for ME. And I'm showing you here the 5 other lab leads that are part of this terrific collaborative group that we have. We just heard a really interesting presentation from Dr. Fair about the itaconate shunt hypothesis. And I won't go through that in any detail. He did it very beautifully already. But just to reiterate that the itaconate shunt hypothesis is that one of these central pathways for generating energy in the mitochondria is the TCA or Krebs cycle. And under inflammatory conditions, such as after an infection, the hypothesis is that the expression of an enzyme called ACOD1 is shunting carbon out of this TCA cycle to create itaconate, and that is depriving the TCA cycle from carbon, which ultimately impacts the production of energy in the cell. And so when I learned about this hypothesis, I got quite interested in seeing if we could test this hypothesis explicitly by expressing ACOD1 and forcing the shunting of carbon out of the TCA cycle to produce itaconate. And then it wanted to test whether that expression of ACOD1 was sufficient to reproduce some of the clinical symptoms of ME. Now, to do this, we sort of had a typical choice in biology, which is choosing between an organismal approach or a cell-based approach. Often in biology, we have to choose between these two choices. And when we do experiments in whole animals like mice or in humans, we have the advantage of having a complex phenotypic system. These organisms we can measure their behavior, their physiology, their metabolism. The problem is, of course, that we can't do very many experiments. It's hard to scale these experiments up and test thousands of hypotheses. So the other choice that people often make is to go with cultured cells, where we can mass produce cells in a dish and then test lots of different hypotheses. This has the advantage of really high scalability, but you lose some of that phenotypic richness. In other words, the ability to look at physiology and behavior and other complex phenomenon. And so my lab has been interested for now a couple of decades in the zebrafish as a model organism because it gives some of the best of both worlds. It's a fully integrated organism with many of the same behavioral and physiological attributes that humans have. But unlike rats or humans, we can conduct experiments on very large scale, enabling us to test many, many hypotheses at the same time. The zebrafish, some of you may be familiar with it. It's a tropical fish, probably sold in your local pet store. It's about an inch long, but it has many advantages as an experimental organism. It has many of the same organ systems that you and I have, very similar physiology, and 82% of the genes that cause diseases in humans have an analogous gene in zebrafish. They're also very prolific, so we can grow them in large numbers in our lab, and each female can lay about 200 eggs a day. So it's no trouble at all for us to generate in a single morning tens of thousands of zebrafish. I'm showing you here in this slide a little vial holding 10,000 freely swimming zebrafish larvae. This enables us to study thousands of animals in parallel and really accelerates our research. One of the kinds of experiments that we like to do is to measure the behaviors of these animals. And what I'm showing you here is a picture from a plate that has 96 wells, and each of those wells is housing about 10 different freely swimming zebrafish larvae. And so in a dish the size of your cell phone, we can house and assess the behaviors of 1,000 animals in parallel. And in my lab, we often build specialized instruments that allow us to track and quantify the behaviors of these animals as they respond to different stimuli or to different drug treatments. So as just an example here, I'll show you a little video of these 1,000 zebrafish. And when I start the video, you'll see that they'll be swimming around spontaneously in the dish. We're monitoring and quantifying all these movements. And then after a few seconds, we'll deliver a stimulus to these fish. And you should be able to see the way that the animals respond to that stimulus. So here the fish are swimming around, and then the stimulus is delivered, and it dramatically increases the swimming rate of these zebrafish as they respond to that stimulus. And now the stimulus has gone away, so they're back to spontaneous movement, and you'll see another stimulus delivered right here. And once again, the animals, almost all, are responding to this stimulus, but we can quickly identify a few wells like this one here where the animals are responding in a different way. And so this enables us in parallel to test 96 different drugs to measure their impact on the behavior of these animals. And with tools like this, what we're able to do is then take normal zebrafish, modify them genetically so that they begin to exhibit some of the attributes of a human disease. We can then array them into the wells of these 96 well plates and measure the physiology or the behavioral changes that occur in these animals. And we can screen thousands of different compounds to try and find those that suppress the disease phenotypes and restore the animals to normal. Those compounds then end up becoming interesting candidates for therapeutic development. And over the last years, we've had several different compounds that have been discovered in this way with zebrafish screens, have been validated in mammalian models like mice and rats, and then moved on to human experimentation. So in the case of the ME modeling, what we wanted to do as a first step was to create a model in zebrafish of ACOD1 expression. This would be a model where we could control the turning on and off of ACOD1 and see whether turning ACOD1 expression would have any impact. And then in step 2, we would characterize this model looking at various behavioral and physiological changes that occurred. And then step 3, if we found interesting phenotypes, would be to conduct small molecule screens to try and find new therapeutic compounds that could be used to treat this condition. So here's a little cartoon showing you the kind of model that we wanted to create. And we wanted to create a transgenic line that had a fairly complex artificial DNA segment in it that had various components. One is that it would have a green fluorescent protein that is driven by a promoter that causes the protein to be expressed in the lens of the eye. The purpose of this would be simply to know which of our animals had the transgene in it. And so that would be manifest by a zebrafish with a glowing green eye, like I'm showing you here on top. But also in that same piece of DNA, there would be another gene, the ACOD1 gene, which is shown here as CAD29. That's this particular variant of the ACOD1 gene that's coupled to a red fluorescent protein called mCherry. And all of that would be under the control of a promoter called the heat shock promoter. This is a little piece of DNA that when it's located near a gene, that gene is turned off until you heat shock the animal. In other words, expose it to warm conditions for a few minutes. And that will turn on the gene and make ACOD1 expression. And so what we envisioned is that the fish would normally be uh, non-fluorescent, but except for their eyes, which would be green. But then we could put them into some warm water, and that would induce the heat shock response and turn on ACOD1 expression in all of the cells. And we would know that it was on because all of those cells would also start to glow red. So that was the idea. I won't go through all the details, but we were then able to use this gateway cloning strategy to build this fairly complex piece of DNA that had the heat shock promoter expressing ACOD1 and the red fluorescent protein, and then this green marker expressed in the eyes. And when we took that piece of DNA and injected it into our fish, we were able to make a line of fish that is shown here. In the bottom, under regular normal light, you could see the fish here with the head to the left, the body, the tail, but it's not at all fluorescent until we heat shocked it. We put it in water that was about the temperature of our bodies, 37 degrees Celsius, and within an hour or two, the entire animal started to glow red, indicating that we had turned on the ACOD1 gene in all of those cells. So the first step was a success, and we developed a protocol where we would take these embryos and once a day for 5 days, give them an hour of heat shocking at 37 degrees, and the expression would come on in the animals where we expected it to, and we knew that we could now test the impact of that ACOD1 expression in these animals. And so the first test that we did was a very simple behavioral test where we would expose the animals in these 96-well plates to, they'd be in the dark for 3 minutes, and then we'd turn on the lights for 3 minutes, and in the dark again for 3 minutes, and the light again. And we kept alternating dark and light. And what we then did was to quantify how much the animals moved in response to these dark and light stimuli. And I'm plotting here at the bottom left the amount of motion that these fish exhibited as it went from dark to light to dark to light to dark. And what you can probably see here is that when the bright light is on, the fish don't move much. They kind of hunker down in their wells. But when the lights go off and the fish are less threatened, they get up and they start swimming around. And we can quantify how much they swim. And we can compare various different conditions. And the most important condition to compare here is the light gray and the red. The light gray are the normal animals that are not expressing ACOD1. And in red, you can see the movement of the animals that are expressing ACOD1. And what we observed was that there was about a 40% reduction in the amount of swimming that the animals that were expressing ACOD1 would exhibit. So that was suggestive that there might be an energy deficit in these animals that was limiting the amount of swimming that they could perform. The second test that we performed was one where we would vibrate the plate that they were in for about 2 seconds. This is kind of a physical stimulus that would cause the fish to swim quite vigorously. And then we would turn the lights off and let them rest for 3 minutes. And then give them another 2-second vibration and let them rest for 3 minutes. And after several repeats of that, we were able to quantify how much they moved in response to the vibration. And again, we're comparing this light gray bar to the red bar. And you can see that the red bar is the animals with ACOD1 expression, and they have a substantially reduced amount of response to the vibration. Interestingly, during the rest period, the difference was even greater. These animals swam very little. The ACOD1 expressing animals swam very little during the rest phase, as if they were having a hard time recovering from that burst of swimming. And so they moved less during the rest period. Dr. Fair also mentioned that in part of the hypothesis would suggest, the itaconate shunt hypothesis would suggest that ACOD1 expressing animals consume less oxygen. And so we used a device called a respirometer here to quantify how much oxygen our normal zebrafish were consuming and how much the ACOD1 expressing animals were consuming. And we started by placing different numbers of zebrafish into this chamber, 25, 50, and 100. And we could quantify the amount of oxygen remaining in the chamber. And as you can see, in all of those cases, oxygen is consumed, and it's consumed faster the more animals you have in the respirometer. We then took a set number of zebrafish and put them in the respirometer and compared normal zebrafish with zebrafish that were expressing ACOD1. And what we saw was a substantial reduction in the rate of oxygen consumption in the zebrafish that were expressing ACOD1. And that was repeated in multiple trials. So like in the human case, we believe that these ACOD1 expressing animals are producing less energy, consuming less oxygen, and moving less than their normal counterparts. We also found that we could, using different levels of heat shock, we could express low, medium, and high levels of ACOD1. And we found that the more ACOD1 we expressed, the more severe the impacts were. At the highest levels of ACOD1 expression, these fish really did not do well. And we began to see decreases in the heart rates in these animals and increase in measures of heart failure developing in the highest ACOD1 expressing animals. So after characterizing these animals a little bit further, we wanted to test whether we could inhibit ACOD1 and rescue any of the defects. And so we've conducted our first experiments in that regard using 4 different compounds that were discovered by one of our collaborative partners here at University of Utah, Eric Schmidt lab and Toto Oliveira's lab. Dr. Oliveira will be giving a presentation after mine that will explain a little bit more about how these compounds were discovered. But what I'm showing you here is the movement of zebrafish first in the first bar with no ACOD1 expression, and then the second bar, animals that are expressing ACOD1. And as expected, you see a reduction in the amount of movement. And now the following 4 bars are fish that have been treated with these 4 different compounds, compounds 1 through 4. And what you should be able to see is that these compounds are increasing the amount of movement in these animals and restoring it essentially to normal amounts of movement simply by inhibiting the activity of the ACOD1 that's expressed in these animals. So that's where we are so far in these studies. We've been able to create this CAD29 plasmid, this piece of DNA that enabled us to create a line of zebrafish capable of induced expression of ACOD1. And we've found that these ACOD expressing animals move less, especially following periods of induced activity. We also found that these animals have reduced oxygen consumption, as would be predicted by the model, and that at the highest levels of expression, they also begin to develop cardiac defects, such as a slow heartbeat. And then finally, we've been able to show that these ACOD1 inhibitors are able to restore normal movement in the animals that are expressing ACOD1. With that, I'd like to point out that Dr. Teja Zhang performed all of the experiments that I talked about today, and that we've had really wonderful discussions amongst our group and with collaborators as shown in this list. And once again, want to just express appreciation to the Khosla family for their gift, which has enabled all of this work. Thank you very much.